Human genome editing is the deliberate alteration of selected DNA sequences in living human cells, and it's become one of the defining technologies of 21st century science and medicine. The science, ethics, and regulation of human genome editing have been the subject of 2 international summits, 3 reports by the World Health Organization, the WHO, a joint report by the UK's Royal Society and the USA's National Academies, and reports by the Nuffield Council on Bioethics. And 2023 will see the 3rd International Summit on Human Genome Editing taking place in the UK. Now, one area that has led to particularly heated debate is germline genome editing, so editing genomes in sperm, eggs, and early embryos. So I'm going to hand over now to tonight's co-chair, Professor Robin Lovell-Badge. And Robin is Chair of Trustees here at PET, and he's also group leader in stem cell biology and developmental genetics at the Francis Crick Institute in London. And he's chair of the planning committee for the 3rd International Summit on Human Genome Editing.
It gives me great pleasure to introduce our first speaker, who's Katherine Littler, who is the co-leader of the World Health Organization's Global Health Ethics and Governance Unit. And she was the person who coordinated the work on the reports on human genome editing that I was part of. So there's 3 linked reports: a framework of governance, recommendations, and a position paper.
It gave me great pleasure to hear that people have confidence that these reports can make a difference. And I guess the challenge for me and my colleagues is the implementation of these reports. And I think the big challenge for me at WHO is the global implementation, because it's not a level playing field. We focused on governance, and the governance is very varied all over the globe. And I think that's going to be a really big challenge going forward, to make sure that as many people reap the benefits of these types of technologies as possible. So that said, just a little bit of background. So the WHO committee was formed in December 2018 as part of the new science division at WHO, really to focus on emerging technologies and the ethical and governance issues associated with it. And it was set up by the DG personally, and it was set up to focus predominantly on governance. We were very lucky, I think, as a committee to, as mentioned in the introduction, there's the International Commission had also begun. So we piggybacked on their work. They focused a lot on the safety and efficacy, and we built on their findings. So we owe a debt of gratitude, I think, to the work they did so that we could build on that. But we had a slightly wider remit than them. And I think it's important to note that, because as noted, germline is where a lot of the ethics debate has been. But I think a lot needs to be said in terms of governance mechanisms about somatic, because somatic is where potentially the greatest health benefits will flow. So getting the governance and the oversight of somatic is also important. And while some countries, I think, are probably ahead of the curve, may think they've got it right, I think there's a lot of countries who don't have pre-existing governance mechanisms for somatic research, and really, in order to benefit, need to move forward. So when it was established, I think the key points to say are really that it was about governance at the global, regional, national level, very much focused on not just heritable and germline, but somatic, for which it did receive some criticism, as some people said, why are you looking at somatic? But I've explained the rationale as to why it did that. The outputs, so I think it's, as I said, the committee was formed of 18 people from across the globe. So it included all the WHO regions, very inclusive, diverse expertise with very different starting points. I think Robin will acknowledge that there were people who came to the table, I think, and had a very different starting point. So getting consensus in a 2.5-year period was a really interesting exercise to do. But I do think there was a lot of common ground, and that comes out in the reports that the committee put together. So as you can appreciate, the work was interrupted by COVID, but I'm going to reflect on the benefits of that when I talk a bit about the engagement and recommendations and future activities. even though we were interrupted, we still managed to have 6 committee meetings, 4 working groups in particular areas, one of which is registries, and I'll come back to that, and I'm hoping we can have more time in the discussion for that. 15 online webinars, like spanning a very diverse range of topics, as you'll see from the recommendations. We had numerous consultations on the registry, 2 satellite meetings and 2 online consultations on the governance framework, one on the very early stages of the governance framework and one when it was much more developed. It's worth saying, and then in terms of the outputs, so obviously I've said the registry, that was one of the very early outputs. I'll come back to that. There was also a very clear policy statement by the Director General of WHO that it would be irresponsible to use genetically altered embryos for reproduction quite early on. And a follow-up statement came in 2020, a governance framework, 9 recommendations for governance and oversight, and obviously all the reports. It's based on best practice in emerging technology, but framing it very much towards human genome editing, but being cognizant of other types of technology, because part of the task of the committee was to really situate their findings in the broader landscape. And that's partly pragmatic. That's partly because there is a growing interconnectedness between different types of technologies. And it's partly, I would say, part of the pragmatic decision. It's easier to be able to set new committees, new regulation, new governance up for broader issues than it is sometimes for narrower issues. So situating it and making sure that whatever was put forth for human genome editing didn't contradict other types of governance mechanisms put forward for other technologies was really important. So the governance framework, as I said, it's got 6 or 7 distinct areas in it. One looking at principles, very important, and actually, interestingly enough, those principles have been picked up in multiple pieces of work at WHO, including one looking at a framework of life sciences, so broadening this out. The work has also been picked up by the Science Council, which is currently looking at the topic of genomics, and they are very interested in the framework. So the principles are being picked up widely, which is good to see, and across WHO, and we are liaising with other UN organisations to pick those up as well. It comes up with a plethora of tools, and I think the tools are really important because it has a broad definition of governance. So it's looking at both the formal mechanisms you can use and informal mechanisms that can be used both by royal societies, both by organisations like PET associations, but then formal legal mechanisms that can be used by governments, including treaties, legislation. It has a series of scenarios to illustrate it, but the proof is, I think, in the pudding. So there's definitely a section on implementation. I want to move on to the recommendations and make a few comments about this. I think one of the great things about the WHO committee is they saw that it is a problem with some of the issues are unique to human genome editing, but some of the challenges with the governance of human genome editing are systems challenges, bigger, wider issues, and we can't just look at it alone in the context of human genome editing. So they decided to write a recommendations document with 9 distinct recommendations to actually address some of these systems challenges. And I think that's where the committee situated its thinking. It really thought, what are some of the underlying issues? And they do come down to research culture, cultural practice. You know, when is it right to speak up if you know something's happening? So I think some of these are the type of research stewardship and culture issues. They are about what can be done where and how, and issues of research, medical tourism, and shopping around in different types of regulatory environments to maybe do something that you wouldn't be allowed to do in one environment to do it in another. So I think the committee thought long and hard about these and came up, as I said, with 9 distinct, 2 or 3 very much focused on WHO, others looking at international research and medical tourism, others looking at what is colloquially called whistleblowing. But I do think that's a really important issue, that we need to think of mechanisms how to do that effectively. And there's been some really interesting work done on that, looking at the stem cell community. And I think the stem cell community, we owe a debt of gratitude. Often when you owe a debt of gratitude, it can be both the successes and failures. And I think they've led the way for some of our learnings, and they led the way particularly for some of our learnings in relation to the registry. So the registry is one of the recommendations and one of the direct outputs of the committee. So early on, we formed a working group, and a registry was set up for clinical trials. So we piggybacked on the ICTRP, the International Clinical Trials Registry platform, and set up distinct categorization for looking for human genome editing. And started pulling out, I think there's some 100 and I've forgotten the exact number, but over 100 trials in that registry at the moment. But what we learned from the stem cell community is by registering trials, some people can actually use this to make a trial look more bona fide in the eyes of the public than potentially it is. So one of the key recommendations of the group was that we should look up at a vetting mechanism. So how can you identify human genome editing trials within that registry that may not be up to scratch, for want of a better phrase? So that's one of the next steps we'll be looking at, that categorization, but also that we need to establish a small group to really look at the criteria for that. And then the third one, and it's interesting to have Kathy on this call, because I had her in when we were looking at this as a recommendation, the committee actually discussed with Kathy the idea of there being a basic research registry. And I think there's a lot of merit in that. And one, to stop duplication of effort, transparency, but also to know what's going on. I don't think it's hard to understand why not to do it. We didn't do it at the time because it was quite difficult with the ICTRP, which is specifically set up for clinical trials, to actually change the categories for basic research. But I am still hopeful that that is something that will be taken forward. And actually, that's maybe something that should be taken forward by the scientific community itself. Maybe they're the best placed people to set up the categories for that. So that's food for thought. And then I know I'm running out of time, but I do want to make a couple of comments on one of the other key themes, which was education, engagement, and empowerment. And it's critical to have all three of those words together. And I don't think the use of education in this context is, I think some of the criticism is it can be belittling, but that's not true. I think the valuable lesson, I was going to say, one of the values of the work being slowed down by COVID is we've also learned from COVID, particularly in the idea of misinformation, the danger of the overload of the information and not being able to get good information out there. And I think for us as a community, that really strengthens the need for engagement, and really good engagement. And I think we should also be upfront and honest. And the committee was really big on this and called for the UN to put together a task force, and we're still working on that. And I think that's an excellent idea. But I welcome all the initiatives in this space. I welcome the citizens' juries, the global observatory, because I don't think it's a one-size-fits-all model. I think we need a plethora of engagement activities.
Well, our next speaker is Kathy Niakan. So she's been my colleague at the CRIC for a while, but we're beginning to lose her as she takes up her new post as Professor of Reproductive Physiology and Director of the Centre for Trophoblast Research and Chair of Reproduction Strategic Research Initiative, all at the University of Cambridge. So Kathy, of course, is well known because she led she was the first to obtain an official license from the HFEA, so to do research that involved editing the genomes of early human embryos in order to study the function of a gene.
There are a number of methods that can be used to edit the human genome. Most of these methods rely on the induction of double-strand breaks to DNA. So DNA is double-stranded, and these methods rely on induction of these breaks using a number of different enzymes. And these enzymes are a type of protein that can be guided to specific locations in the genome that induce a break in a targeted fashion. And after the induction of a double-strand break to the DNA, repair machineries that exist in all of our cells are activated and help repair the damaged DNA. And we can take advantage of these methods to introduce mutations, to inactivate, for example, the function of a gene, or to introduce precise genomic changes to DNA. And a method that uses the Cas9 enzyme, which is shown here in teal, is currently the most widely used and efficient method to edit the human genome. And this is because it's comparatively much easier to program the enzyme to target specific DNA sequences in the human genome. So this enzyme Cas9 can be guided to target sites using an easy to modulate guide RNA, which is shown here in blue. And the enzyme then induces a double strand break to the DNA, and repair of the break can be error prone, so that can lead to a loss or gain of DNA nucleotides. And so we can take advantage of the error prone method to repair DNA damage, to make target genes non-functional. So this allows us, for example, to understand the critical role of a target gene in early human development, to understand whether it does or does not have an important function in this critical window of development. The Cas9 enzyme itself can also be modified, where instead of the induction of a double strand break to DNA, the enzyme instead nicks only one strand of double-stranded DNA. And this allows the introduction of point mutations in DNA without the generation of double strand breaks. And this is a method that's called base editing. However, base editing has been shown recently by a number of groups to lead to what are called off-target edits to RNA, and not just the intended DNA target sequence itself. So genome editing tools can be introduced into human embryos using either microinjection into eggs, coincident with sperm injection and fertilization, or at slightly later stages of development, for example, in fertilized eggs or subsequent cell divisions. And there's also a possibility of targeting egg and sperm cells before these stages, or indeed precursors of these cells. And I think in the future, this may be perhaps a more favorable method that's easier to screen. So for now, genome editing methods have been introduced into either viable or non-viable human embryos. And this is just a chart summarizing all of the publications so far targeting human embryos using genome editing methods. Most groups focus on achieving correction of mutations associated with human disease to introduce designed edits into the human genome as a proof of principle for clinical applications. However, I think it's clear that many technical limitations still exist that make precise editing of human embryos difficult using existing methods. And this is evidenced by the generally low efficiency of mutation repair to date. And in addition, there's consistently an issue of what's called mosaicism, where not all of the cells of an embryo have undergone genome editing as intended. There's also the clinical use of genome editing by He Jiankui, which is summarized here, and this is the data that was summarized from what was presented at the International Summit on Human Genome Editing. And the data suggests that the embryos were mostly mosaic, and that the efficiency was not 100%. So there were issues in both regards. Base editing from one group, from one study, suggests that it's perhaps much more efficacious in terms of targeting efficiency, but this has not been replicated and by other groups. And I think it's important to stress that evaluation for the possibility of off-target RNA editing has not been performed, and that could be a major issue. So we used genome editing methods in our research, principally to understand early human development. And we inactivated a protein that's called OCT4, which we predicted would have an important function in early human embryos prior to implantation in the uterus. And what we discovered is that without this OCT4 protein, human embryos arrest at stages where many embryos in fertility treatment also fail to develop. So that teaches us a critical role for OCT4 protein in functioning to allow for embryos to develop to this important stage where they would be ready to implant. We used what's called whole genome sequencing to map genomic DNA sequencing reads to chromosomes, and this allowed us to determine that in most of the genome edited embryos, there were discrete small mutations that inactivated the target gene OCT4. And that allowed us to recover 2 copies of chromosome 6, which is where the OCT4 gene is located. However, in some cells, we found structural changes to the chromosome that led to a segment being either lost or gained, and this suggests possible complexities that I think are really important to consider in both basic science and in thinking about potential clinical applications, not just in human embryos, but also in human somatic genome editing. And we can now screen for these complexities in basic research. And this is consistent with a series of publications that report on similar large deletions or genomic rearrangements in other cell types using the same Cas9 enzyme, but different guide RNAs. Okay, so just as a summary, our work and those of others suggest that a number of uncertainties and challenges remain, including improving rates of precise edits, the issue of high mosaicism, where not all cells of the embryo have been similarly edited, is also a persistent issue, and similarly edited in edited embryos on or off-target effects are also important considerations in the future. So ultimately, I think it's really important to stress that more basic research is needed if these methods or any derivatives of these methods are to be applied clinically in any context, really.
So I'll move straight on to our next speaker, who is Professor Reik, who's the founding director of the Cambridge Institute of Science at Altos Labs, but he's also still a principal investigator at the Babham Institute. And he is an honorary professor of epigenetics at the University of Cambridge and the Cambridge Stem Cell Institute.
There are at least as many epigenomes probably as there are cells in the human body, which is 200 or 250. But probably there are more epigenomes because they're also susceptible to external conditions, environment, nutritional influences, and as I mentioned, also changes during aging. So there are many hundreds of epigenomes in our body. So what are epigenomes? So they're characterized by modifications of the DNA in the form of, for example, DNA methylation or other chemical modifications to DNA, how the DNA is wrapped around the histones, how histone tails are modified chemically themselves, and how these epigenetic modifiers and factors contribute to modifying how the DNA functions basically in cells and organs and in the body and over time. And so very simply sort of speaking here is that epigenetic marks of this kind of nature can affect transcription and other genome functions, DNA replication, DNA repair, other genome functions. Epigenetic regulation is very important in health and in disease. Many mutations in epigenetic modifiers have been detected in human diseases, cancers, etc. And epigenetic modifications can be somatically heritable in somatic cells in the body, but they're actually erased in every new generation as a new embryo develops. But this is not the case in plants, and I will comment on plants at the very end of the short presentation. So what can we discover in terms of epigenetic regulation in the genome? So there are modifications to the DNA, as I was saying, DNA methylation, other DNA modifications. There are modifications of the histones, there are transcription factors that activate or silence transcription, non-coding RNAs that may contribute to this kind of process. There are things that bring things together in the genome to affect gene regulation at smaller scales and potentially at much larger scales in the nucleus, bringing things to the periphery of the nucleus or to the center of the nucleus, and thus affecting gene expression programs. So drilling down a little bit more into the detail of these fascinating epigenetic questions, and what can we achieve by epigenetic, what can we understand better by epigenetic editing approaches. So for example, we could bring different parts of the genome together experimentally by editing to ask what's the function of DNA topology and organization in the nucleus. We can understand gene regulation better by modifying or editing DNA methylation locally, histone modifications. We can understand better the significance of the non-coding genome. Remember that there's only 20,000 or so genes in the genome. The rest is non-coding. Is a vast majority of the genome. Or does that work? How can we edit this? Epigenetic processes can confer memory on cell divisions, how cell fate is maintained in the embryo and in the adult. And then we could also, by epigenetic editing, simplify complex relationships. For example, we can put multiple edits into the same locus by histone modification, transcription factors, all at the same time. So there's a huge sort of potential there to change things epigenetically in the genome. So just briefly to comment on this sort of expanding toolbox of what is possible. And this is actually, so the basic is very simple, and it goes back to what Kathy just beautifully explained on the genome editing. So instead of active Cas9, we use inactive Cas9 basically. And that's to bring the any effector, like an epigenetic modifier, editing molecule to the DNA. We use what we call dead Cas9, so it's not active in cutting the genome, but just brings effectors to the genome. And this could be things that methylate DNA, demethylate DNA, histones, etc., transcription factors, etc. Then we can string multiple effectors together using so-called SUNTAC, whereby you can put little effectors together that edit locally DNA modifications, histone modifications, bring transcription factors to the same place. We can do the same with an RNA kind of scaffold here, where, for example, we can bring multiple transcriptional activators to the same location in the genome. We can miniaturize the system, and this can be quite important for delivery, for example, viral delivery of these kind of systems, where you have only so much space in your viral delivery molecule to deliver to the cells that you're interested in. Then you can split the system, whereby the two components here, You can bring them together by light or by chemicals. And that brings, for example, if you put these things into different places in the genome, then all of a sudden you can bring together different chromatin or different DNA molecules in the genome and ask what could be the functional consequence of this. Then we can also use, instead of Cas9, we can use Cas13, which can modify RNA molecules, not only DNA molecules in the system. This is just a very brief sort of snapshot of ongoing work in our lab by Corinne, Melanie, and Chris in the lab. They put together this very potent combination of transcriptional repressors, and they show that this works very nicely on different promoters. It's more challenging to work on enhancers, actually, so you have to try harder to inactivate epigenetically enhancers in the genome, but putting these very potent cocktail of inactivating systems together, they've managed to very potently inactivate some particular enhancers in the genome as well. So how can we think about using these systems in potential therapeutic settings? This, at the moment, this is in animal model systems, but a number of very interesting and successful interventions have already been carried out. So, for example, you can have haploinsufficiency in certain diseases. You can activate the silent allele. You can have dominant acting mutations that you can repress epigenetically. Then you have epigenetic diseases, such as, for example, Fragile X or imprinting diseases. You can modify the epigenetic makeup of the other alleles to sort of reactivate silent alleles, for example. And you can begin to think about complex disorders.
The next talk I'd like to introduce is from Hank Greely. So Hank is the Professor of Law and Director of the Center for Law and the Biosciences at Stanford University. He is the author of several books, including CRISPR People, Science and Ethics of Editing Humans, and The End of Sex and the Future of Human Reproduction.
So let's start with somatic cell editing, which is fundamentally another way, one more modality of gene therapy. Gene therapy was first tried arguably about 40 years ago. It's finally meeting some success. Like many things in biomedicine, it's turned out to be more complicated than we expected. I don't think genome editing in the somatic cell context poses special ethical issues. It has all the ethical issues of any new biomedical technology, the most important of which often aren't thought of as ethical issues, but they're profoundly ethical issues: safety and efficacy. It is unethical to do unreasonably unsafe things to people, and it is unethical to do things to people that aren't going to work. Safety and efficacy are, I think, far and away the most important points. There are also points more salient, I'm afraid, in an only semi-civilized country like my own, which doesn't cover healthcare for everyone. But the access to these new therapies, the prices of these new therapies also raise significant issues. I do think that the big safety and efficacy question with somatic cell is going to revolve around rogue clinics. I'm fairly comfortable with the FDA, the EMA, the various national authorities that have jurisdiction over this to do a good job. But as we've seen in the stem cell world, that doesn't stop rogue clinics, even in countries with strong controls like the United States, from actually popping up everywhere. So to me, that's a big ethical issue to watch with somatic cell genome editing. I want to move to germline, although in doing so, I want to point out that that distinction is not as clear as we sometimes think about it. So when we say germline, probably because of Hu Jiankui and his fiasco, his scandal, we think of embryo editing. We're editing the embryo, and editing the embryo, if it's done right, will end up with every cell of the resulting human being edited, including the germ cells, the eggs or the sperm. Now, you could have embryo editing at a different point in embryonic development. We don't change names from embryo to fetus until about 8 weeks of development. And you could, in theory, do an embryo edit that didn't involve the germ cells after the germ cells had already been segregated out from everything else. Or alternatively, you could do a somatic cell edit in an existing human that does involve the germ cells by changing eggs or sperm or the genes or the cells that give rise to eggs or sperm. So the line between somatic and germline is not quite as simple as people make it out to be. But for present purposes, we're fundamentally usually thinking about embryo editing when we talk about germline editing. I don't think embryo editing or germline editing is going to be as important as people think in two different directions. First, I don't think there's some deep principle involved here which cannot be violated. It should not be violated. I don't think, there are people who seem to act as if the human germline genome is the Ark of the Covenant, is the essence of our species, is sacred, it must not be touched, and certainly must not fall into Nazi hands, if you've seen the Raiders of the Lost Ark. There are 7.8 billion human germline genomes. There are no two people who have exactly the same human germline genome. Even identical twins have subtle differences based on somatic mutations after they split. The embryo that became both of them split. The genome changes with every generation because of mutations during the lifetime of the parents. I don't have exactly the DNA that my parents got from their parents. My kids don't have exactly the DNA that I got from my parents. For the most part, those differences are unimportant. Sometimes they will be important. And then human activity actually helps select what the human germline genome is going to look like. Agriculture has changed the genome. Eating more starch gave a comparative advantage to people who had more copies of a gene that helps digest starch, and gave more advantage to their children who inherited extra copies of that gene. Medicine changes the genome. The discovery by Banting and Best that they could take insulin from pig and cattle pancreases from slaughterhouses and keep alive people with type 1 diabetes who otherwise would never have reproduced because they would have died before they could reproduce, means that they're now type 1 diabetes isn't strongly genetic, but there are some significant genetic associations. There are undoubtedly more copies of risk genes for type 1 diabetes now than there were 100 years ago, because people with type 1 diabetes, including people who have those risk genes, now can reproduce successfully. My cousin Mike, who died last year at 69, had type 1 diabetes from the age of 8. He had 16 grandchildren, none of whom would have existed before medicine intervened and changed the overall shape of the human germline genome. So I think that the human genome, individually and collectively, something we need to be careful about, but it's not a sacred object that's unchangeable and has never been changed by humans. And frankly, in general, I prefer conscious, knowing, intelligent, carefully made changes to those that are kind of random. On the other hand, I don't think there's very much good that human germline genome editing is likely to do. Most of the things we can do through human germline genome editing, we can do through pre-implantation genetic diagnosis in terms of avoiding monogenic traits, particularly diseases. We are increasingly able to do through gene therapies or even small molecule therapies. We're getting much better at treating many of these genetic diseases. I think there is a use case for people who, say, a couple where each of them has the same autosomal recessive disorder. The only way they can have kids that are genetically, quote, theirs, close quote, who don't have that disease is to edit their genes. So a couple who both have cystic fibrosis, they live long enough, they're healthy enough, they fall in love, they want to have kids together, but they don't want their kids to have cystic fibrosis. They've survived, but that's not an easy or pleasant disease to have. For them, human germline editing would make sense, could make sense, depending on what the alternative treatments are at that point. But there aren't going to be very many people like that. So I think the big fear many people have of germline editing is super babies and the caste system and speciation events, all of which makes for great science fiction, none of which we know how to do. I mean, in order to make super babies, you not only need to know how to edit things, you need to know what to edit and in what ways. And we know effectively nothing about genetic variations that enhance people, let alone that make them the X-Men or other wonderful mutant superheroes. So maybe our grandchildren will have to worry about it. I'm willing to let them worry about it. I don't think we have to worry about it for the next several decades at least. A couple of miscellaneous items to end. First, I was really pleased with the WHO for its position about whistleblowing. I'd argued for this, although in order to get attention, I had called it, in an American book, I called it snitching. I think for you Brits, it would be grassing, if I've got the lingo correct. It is a really interesting phenomenon and one that is culturally discouraged, but culturally important. There are some ways that people have come up with methods that make this more effective. And I think science needs to aspire to it. Scientists need to feel a duty to report badly unethical science, but the devil will be very much in the details. And I hope WHO continues on with this at length. Second, I think we, and by we here, I mean particularly those of us who work on ethics, need to reconsider some of our issues about consent and human subjects research. I think babies born as a result of human germline genome editing need to be research subjects, need to be mandatory research subjects. This goes against our general rule that we should only make people research subjects with their consent. Now, parental consent we could have, but then when the kid gets old enough to assent, at some age, I think the child should be able to pull out, but when? We really need that information. I think if people go into experimental trials, they should be able to commit their children to X years of research follow-up. It's so frustrating that after, 43 years of IVF, we still don't have as good information about health and safety as IVF as we'd like. We know it's pretty healthy, it's pretty safe, but the registry is a good idea, but I think we need to rethink mandatory participation in the registries and in follow-up research.
So finally, this evening, we have Nick Meade, who is the Joint Interim Chief Executive and Director of Policy at Genetic Alliance UK. He's also co-chair of the Rare Diseases Coalition.
So my first point is there are a lot of rare conditions. The EU and the UK define a rare condition as affecting fewer than 5 in 10,000 people. Almost every single gene condition is rare. And we've got about 6,000, just over 6,000 of them on the OrphaNet database, which is a great site to see more about rare conditions. About 72% of the ones there are genetic. So if I turn to, so I've just said there are a lot of rare conditions. There are not a lot of rare disease treatments. The International Rare Disease Research Collaborative is very generous in the way it counts medicines for rare conditions, and it lists 393 new medicines in the previous decade for rare conditions. I think most regulators would count that more like 200 or so. But in the same period, 848 unique new rare conditions were added to the OrphaNet database. So we're not keeping track there. There are other treatments as well, such as bone marrow transplantation, and dietary supplements for exclusions and so on. But the burden of treatment and or the risk that people take is really usually extremely high for rare disease treatments. I'm going to move on to reproductive choice, which at the moment, certainly if you live in the UK, is the best chance you have of taking control of the way a rare condition affects your family. I'm going to accidentally call it PGD at some point, but PGTM, pre-implantation genetic testing for monogenetic conditions, is now available, licensed in the UK for more than 1,000 rare genetic conditions. So that dwarfs the availability of medicine in the UK. We also have access to mitochondrial replacement therapy and to non-invasive prenatal testing as well as tools. So the combination of those 2 points, that there's not a lot of rare disease treatments and there are a lot of rare conditions, equals high unmet health need in rare conditions. And that influences our attitudes to a lot of things in health. And so in that sense, genome editing isn't actually particularly unique to us. We've got a relatively permissive attitude towards sharing of identifiable data and the use of that data by a wide group of stakeholders, providing it takes us towards progress to meet this unmet health need, and with the same purpose, a really permissive attitude compared to other populations for the use of animals in research, for reproductive choice techniques, as I've mentioned, and towards examining innovative medicines. So, and that's all in the spirit of leaving no stone unturned in the search for cures, treatments, and other means really to take control of how rare and genetic conditions affect our community. So just to emphasize, genome editing isn't the only stone that we're looking to turn over here, but it's certainly one, as we've heard in the presentations today, that have a great deal of potential. And one of the things we did with PET was ask people what they thought about genome editing, and there were some, you can see this in the reports we've done, but some powerful quotes here. I feel any person with a life-changing or life-limiting condition should be given the chance of a normal life. And again, if it was for the greater good, then yes, I'd be pleased to help this research. And notice the generality of those points that could apply to things outside of genome editing. So I want to use the rest of my time just to reflect on a few of the standard kind of issues that come up in talking around genome editing. The first one, just kind of compare those to how we've seen the same topic come up in other areas of rare conditions. The first one I want to pick up on is equitable access. And I think, first of all, I absolutely am very interested in equitable access. I'm interested, though, in how often it comes up in talking about access to genome editing technology, because there really isn't equity and access to any rare disease medicine or therapy between or within countries. And in the UK, just an example I know most about, there's an equity between classes of condition. You see cancer prioritized in policy and investment, often between countries of the UK with different medicines available. Within conditions even, we see different availability for the same medicine depending on your symptoms, and in access to reproductive choice as well. Depending on how many children you happen to have had in your past, you may or may not be able to have PGD with your current partner. And so I think that's worth understanding just when we start thinking about that. That's not to say that we shouldn't try for equity there, but it may be quite difficult. So the other thing is kind of which conditions would we use genome editing technology with? And this again applies to both somatic use and germline use of genome editing. And I want to just flag, so the 1,000 of conditions that are available for PGTM in the UK have all passed a seriousness test. And all the medicines that we're funding in the UK as well pass a kind of a severity test. But NICE and the Human Fertilization Embryology Authority do that kind of examination in different ways, and with differing success in the view of our community. I think that the key point that I want to make here is you need to think really carefully before you say this rare condition is a severe one or a serious one, and this one isn't. And we want to focus our use of new technology on these conditions because insert discussion of symptoms. And there are lots of aspects to a condition: age of onset, rate of progression, whether it's acute, chronic, or episodic, and how it affects families. Is it a genetic condition that passes down through generations and so on? And actually, if you consider any one of the options I just listed, a condition that affects children at birth, or a condition that strikes in adulthood, the more you delve into the life of that family, the more you can see that that's a serious condition, whichever way you look at it. And I think I'm going to finish just with that point, that if you examine rare conditions and look in detail, then the more you understand how they're affecting people's lives, the more you can grasp the severity of them. And I'd encourage you, it was Rare Disease Day on Monday, to check out the videos that are on the Rare Disease UK YouTube channel we put up for this year, which are really representative of how people living with rare conditions live. We've got Samina, mother to Armar, who sadly died aged 13 to arteriovenous malformation. We've got David, who goes by Mr. One-Peck, who's become a fitness influencer despite his Poland syndrome. We've got Jace, who had to wait until they were 18 for their diagnosis of Nemeline myopathy. And we've got Melissa, an artist who lives with idiopathic intracranial hypertension. And if you look at those 4 stories on YouTube, they're short, then you will hopefully grasp how the more you understand a rare condition, the more you can understand it's serious.
So you apparently made a comment that epigenomes are heritable, yet erased in each generation. And I think that is a point I think that might need clarification.
Thank you, Robin. Yes, absolutely. Sorry, I had to be quite brief in the explanation. So epigenomes are heritable in somatic cells in the current generation. But when it comes to the next generation and sperm and egg forming the new zygote, the new organism, then all these somatic or gametic epigenetic modifications are largely erased. And this is important for the new embryo cells and the stem cells to develop again and to form all the new organs, new tissues, and the new organism. So, and this is a big difference between plant, as I was briefly explaining, plant and mammalian, say, or animal development, is that in animal development, we have this enormous erasure of epigenetic information at every generation, but in plants, we don't. And that gives rise to the potential in plants for cross-generational or transgenerational epigenetic inheritance, which is largely absent in mammalian organisms.
Okay, I hope that makes it clear. Thank you. So we have a question from Eugene Diana, which I think maybe will go first of all to Kathy, but others might want to address this, which is, does the fact that we apparently don't know how many human genes there are, although I can paraphrase that by saying there's a lot of uncertainty about the human genome and I guess genetic variation. Does this affect the relative confidence of doing any treatment-related genome editing without collateral damage?
So yeah, I think as Wolf really beautifully presented in his talk, there are a number of promoters or enhancers, so non-coding regions of the genome, or even non-protein coding regions of the genome that should they be targeted or affected, could affect the function of genes in ways that we can't predict. So that's, I think, would stress the importance of, if one were to develop a method, and there's not one that's been developed, in my opinion, to date, that really confidently says one way or another in the germline context, but if one were to develop that method, you would have to rigorously evaluate this. So methods to do that would be, for example, whole genome sequencing at really high depth in potentially cells that are not embryos themselves initially. And that's where I think maybe the germ cell genome editing might be a context in which one can screen these potential on or off target effects much more, I think, confidently before they could be used. I think that's probably would be my prediction, is if it were to be used, that's probably the context in which it would have the highest chance of kind of avoiding the collateral damage.
So on that, how good is whole genome sequencing in terms of how many cells do you need to get a reliable whole genome sequencing, and then it's not reliable anymore because if you have mosaicism.
Yeah, absolutely. So at the moment, yes, you'd need several thousands or hundreds of thousands of cells. And at the single cell level, we did try to do 30x whole genome sequence, and it's very, because single cell whole genome sequencing relies on amplification of the genome in a way that makes it such that there are gaps in the genome that you can't actually sequence across. So you cannot get that at the confidence at restricted number of cells. I don't know if maybe.
Wolf, do you have any comment on that?
No, I think that was very well explained, Kathy. That yeah, there is a problem. I think that future methods, I'm sure, will improve on single cell whole genome sequencing, but it's not quite there yet.
Yeah. I mean, there's still parts of the genome that aren't sequenced at all well.
Yeah.
Y chromosome being one of them, but that's a special chromosome. Okay. I'm going to jump to a question from Rimiana Yotova, which is particularly for Katherine Littler. Is there any momentum for adopting a formal governance tool, such as an international treaty for the regulation of human genome editing, before it goes to the stage of clinical research too much? Any interest in the states shown in this? And if so, is the WHO the best international body to serve as the platform for those discussions?
So as Robin well knows, when the committee was first formed, I think one of the issues on the agenda was the discussion around treaty and whether this was the appropriate mechanism. And actually in the discussions and in the engagement, this wasn't raised as the number one mechanism at that point in time. And it would be interesting to know, I think now with all the pandemic treaty discussions, whether there will be more uptake for a treaty. But in our deliberations and actually following up from all the webinars that we've done with member countries, the issue of a treaty hasn't been raised. Support for local governance mechanisms has been raised quite a lot, and how we can think about collaborating across regions in formalizing government mechanisms has come up a lot. But the actual treaty, and I'm surprised by that discussion, hasn't really evolved since the early days. But I don't know, Robin, if you want to comment on that as well.
I mean, I was thinking today, because there was news about them starting a proceedings to have a treaty about plastic waste, and 200 countries signing up to try and deliberate on this. That's an obvious one. We don't want plastic waste. That's a really obvious one. But when you're talking about human genome editing, it's not such an obvious question. There are good reasons for having clearly somatic genome editing, and there are one can make arguments in certain circumstances for having heritable genome editing, as Hank said. So I think it's a harder one to think of how you could possibly have a big international treaty that's governing all of this, such a complex area. So, Hank,
I think that blurred line is what Hank was saying about the blurred line. Yeah. How would you define also what you're doing? And I think that appetite question is really interesting. I think the countries were much more interested in the mechanisms that they could put in place, coordination and the collaboration that they have regionally as a better mechanism for doing this particular area than a treaty. So I think you're right. I'm not sure if it lends itself as the best area for a treaty going forward.
Yeah, Hank?
Yeah, I'm not optimistic about a treaty as a mechanism for this. Lots and lots of years of negotiation. There are different interests in different countries and different levels of concern in different countries. In many countries, this really isn't an issue of much concern because they don't have the infrastructure to really be involved much in it. And I think what the best you might get is a relatively vague and meaningless statement that then gets implemented or not implemented by different countries. The Council of Europe back in the 90s, I think, passed a created a treaty within the Council of Europe banning human cloning, but it left the definition of human cloning up to each individual country. And some countries defined it broadly, and some countries defined it narrowly, and many countries didn't define it at all. So that's what I'd worry about, that you're likely to get, if any sort of treaty, one that is relatively vague, ambiguous, and not very helpful. Having said that, though, I mean, I do think there is a role for international collaboration here, but I think it's through things like scientists, physicians, patient groups, and others coming up with guidelines, guidance, principles that wouldn't be legally binding, and yet can often have a significant influence. So things like the WHO effort and the National Academies, Royal Society, et cetera, effort, I think those are powerful and meaningful. The ISSCR, International Society for Stem Cell Research, guidelines in stem cell work aren't binding, but they've had a major influence on how it's done all over the world. So I think looking internationally is a yes. Treaty, not a good way to go about it.
So this is a question from Martina Cornell, originally addressed to Katherine, but I'm going to actually give it to Nick, which is, to what extent are patient organizations now involved in the education, engagement, empowerment aspect of all this?
Thanks for the question, Robin, but I think that probably specifically to Katherine's education, engagement, and empowerment, I will say a few quick points though. Just so we're looking at getting gene therapy into the NHS at the moment, and just to illustrate the need for different information and information to be tailored towards the people you are giving them, the choice that parents of a child with spinal muscular atrophy are making is quite different from the choice that an adult living with thalassemia is making when they decide to take the choice and take the risk to consider using a gene therapy. And I just wanted to highlight the kind of, you need to be situating your treatment in the lives of people living with conditions and consider how their existing treatment affects their decision to use these kinds of therapies. But Katherine, I'm sure you'd like to come in as well.
Yeah, happy to. Thanks, Nick. I mean, I think what really struck me in the deliberations was when the committee went to South Africa and engaged with the sickle cell community. And also I know from previous interactions in relation to the H3 Africa initiative, actually the sickle cell patient community has been instrumental in education, engagement, and empowerment in that setting, partially from an absence of education through more formal mechanisms. So they have really brought up knowledge of genomics, knowledge of human genome editing, and the potential benefits. So I think there is a huge role for patient groups to play in lots of different settings, and to recognize where those groups need to be supplemented by other voices. And back from working with Nick and colleagues back in the UK, I think you cannot underestimate the necessary need for getting these voices, not only to the general public, but the education value of policymakers and decision makers that patient groups can play in getting an issue at the forefront of the agenda. So I think they're a critical stakeholder group in this work going forward.
Thank you. Anyone else want to comment on that? I mean, I think the patient voice is so important. As Nick was alluding to, but maybe also to Hank, that, if, let's say that, I make this point quite often, so I'm going to make it again, that somatic genome based therapy, gene editing based therapies work well, conventional pharmaceutical methods work well, so you get many more individuals with genetic diseases surviving to reproductive ages, wanting to have children. Isn't that going to put yet more pressure on the whole idea of heritable genome editing?
more pressure in terms of,
meaning there'll be more individuals demanding that they can use this to avoid their children having genetic disease, partly because there's going to be more individuals who are homozygous for genetic mutations, etc.
Right. I mean, that's a really interesting question, and I think that's an empirical question that we can't answer right now. It depends. To the extent that treatments for the genetic diseases become effective and safe and easy enough, then why bother with the human genome editing? On the other hand, if they do lead to an increase in the number of people with the risk factors, with genetic risks, then there are more potential uses for genetic editing. How those two different vectors will sum, I don't know. I do think the genome editing is always going to introduce one more complication, one more chance for things to go wrong. And whenever humans are involved, mistakes get made, things go wrong. And so avoiding it has some value just as a result of simplification and having one less thing that can go wrong. I do worry a little bit that I saw something worried about the sort of load of the disease load that we've got. Well, medicine in particular, and civilization in general, works by trying to make things easier for us that are otherwise difficult. And your genome is really only important in terms of how it affects your phenotype in a particular environment. And if it's an environment that has good, accessible, effective healthcare, does it really matter that you've got a genetic predisposition to something that would be a problem if you were put into a wilderness? I don't think so. But it is an interesting question, Robin, as a matter of sort of consumer choice and politics, how that will play out.
Yeah, I mean, cost is going to be another factor given how much current techniques. Nick, you wanted to make a comment on this?
Yeah, thank you. I think as we see treatments for things like cystic fibrosis becoming more powerful, we will be having couples with rare conditions, with recessive conditions for the first time. And currently, people with cystic fibrosis don't meet up because they don't want to share an infection with each other. But now this will be possible. And of course, they won't be able to use PGTM to conceive an embryo that doesn't have CF. So I could see that happening. And then to Hank's point, but if the treatment works, why would you bother? I think we can see that happening in PGD now, that even with relatively effective treatments for rare conditions, families are putting their energy into making sure the child that does have the condition is treated, but they don't have the stamina to have another child that needs that treatment. And however effective a treatment is, it will almost always come with a burden or a risk or both. And a gene therapy, a curative gene therapy at a young age at the moment is quite a big risk with current technology. And some of these treatments that are really very powerful still require good adherence through teenage years and so on. And we do see couples currently choosing to use reproductive choice in those scenarios.
If I might just quickly add, I agree with you, but of course, PG whatever, the proliferation of different acronyms for this technique has surpassed my capacity to remember all of them. PG whatever also is not without its own risks, its costs, its discomforts, and it's it's certainly not 100% effective in terms of, the number of live babies you get per round of IVF, and PGD is not 100%. So there are costs and benefits on all sides, and it's not just financial costs, it's the emotional costs to the patients and the physical risks that they'll undergo. How it plays out will be interesting to watch.
Yeah, in fact, Guido de Wert raises a question about this very topic. So there are instances where you may have to screen for more than one gene variant. So there are, or save your siblings would be a classic case of that. For example, where PGT M or whatever becomes very inefficient, and so correcting one allele maybe would then allow you to allow those parents to have a genetically related child, which is the big issue. Let's move to a different type of question. Katherine, on for you again. How do we ensure education, engagement, and empowerment, as well as ethics, in regimes with state-controlled media, such as Russia and China?
So I would start by reiterating what I think the committee came up with, and that is we need to rely on different models in different settings and different circumstances. That said, the committee also, and I wanted to go back and check, I can't remember how much they put in the report, but there was a really important section about us ensuring that education engagement is not just about the general public, but it's education to policymakers. And I think a classic example of educating policymakers would be getting valuable information to ministries of health and ministries of science and technology. So in this type of setting, that would be critical as a starting point to make sure valuable information had got through them. I think this is another area where we would want to piggyback on the work of our colleagues at WHO who are leading the infodemiology, I find that word hard to say, project. So they're looking at the science of managing the infodemic that's happening. And they're on I think they're on their third international workshop, and they've set up mechanisms for looking at social media across the globe and messaging. And I think we could definitely piggyback on that work in relation to human genome editing as a means of education and engagement and ensuring we get the right messages out there. But then I think this also ties into the science culture issue when you alluded to, would this happen in certain countries? I think it is about the whistleblowing and other practices, but we need to take a very diverse approach to engagement. What engagement looks like in the UK, which has been arguably very effective, you wouldn't be able to have those models in many societies. So I definitely think a lot more work on this, but I'm very keen to look what we can do with our colleagues in relation to emerging technologies and epidemiology, and work with our country counterparts on getting briefings to ministries so that they have the right information in front of them to hand.
Anyone else want to comment on that? I mean, there are efforts in some of those countries to do public engagement properly, and it's doable. It's challenging, but doable.
I guess I'd just say I do think that we are not going to see a uniform global response to this technology. Different countries will have different takes on it, different opinions about it, different uptake, different technological abilities, different healthcare spending systems. There will be variation from place to place, regardless of how good or bad the public engagement and disinformation and what the infodemiologists tell us. I'm not sure I like that word.
No, I don't.
Right. Gosh, where to go next? Well, more practical one to Kathy from Conor Ross. From your results of the Oxford paper, the level of mosaicism, can you realistically see CRISPR-Cas9 technology being used in an IVF clinic? And do you envisage that this technique will be formed by clinical embryologists or scientists?
I mean, not in the version that's been used. No, absolutely not. But it will be interesting to determine whether as methods to establish in vitro derived egg and sperm develop, whether that might be a context in which some variant of the methodologies that were described by either Wolf or myself could be applied.
I mean, the main advantage there is that if you're working with cells you can grow in culture, you can screen cell clones for exactly what you want and avoid what you don't want.
Exactly.
As long as the cell culture itself doesn't cause a problem. Hank, sorry.
That was a major part of my other book, The End of Sex, in vitro gametogenesis, making eggs and sperm. And although the human efforts are still well short of even a proof of principle, let alone proof that you can do it safely, I think this is likely to happen. And its benefit for genome editing, germline editing, is really a side effect. There are just so many couples in the world that would love to have kids and can't because one of them doesn't make gametes, or they both make gametes, but they both make the same gametes. And maybe you could use this to make eggs from men's cells, sperm from women's cells. I think this is something that's going to happen. Whether it happens in my lifetime or not is sort of 50-50 at this point.
Well, I hope you live long enough to see it.
Well, me too. But I'm not counting on another 50 years.
Okay. Wolf, do you have any comments to make on that? Or?
Not really. I think maybe worth thinking about in view of what Kathy's sort of experiments are showing and others, whether efficiency is different between epigenetic and genetic editing methods. That's certainly worth reflecting upon, but I don't have an immediate answer on it.
Also, one question mark about in vitro derived gametes is the epigenome and what's going on there, because as you are, establishing and wiping out imprints, it's a very sensitive cell type to be working with in culture. So, from what's been published, particularly in the mouse, do you think it's there yet, or it's still quite inefficient, as far as I understand?
I think it's still quite inefficient, and as you say, Robin, it's still not completely clear in terms of the normal processes of epigenetic remodeling, what is properly recapitulated in the culture systems. So this will be important to monitor exactly then how, of course, how genetically viable these gametes are, but also how epigenetically viable these in vitro produced gametes are. Very important.
It's interesting on the epigenetics is really important, mysterious, and will be interesting to watch. The relative success, I'll put in scare quotes, of livestock cloning, of cloning in other mammals, is some evidence healthy animals without the traditional egg and sperm and epigenetic rewipe that way. Whether that would apply in stem cell derived gametes, unclear. And certainly something that would have to be nailed down before this was ever used in humans.
So Marcy Donofsky asked actually a couple of questions that are sort of related, but so I guess I'll get Hank and Katherine to think about these. So the Oviedo Convention, the Council of Europe's Convention on Human Rights and Biomedicine, is very clear about its prohibition on heritable genetic modifications. And Marcy says that the Council of Europe has recently reaffirmed this article of the treaty, which is signed and ratified by 29 countries, meaning that treaties can exist. And then there was a paper published which Marcy may have involved with, but Francois Baylis, who was on our WHO committee, was part of too, or one of the authors of, which pointed out that something like 70 countries currently prohibit human heritable genome editing. So any comments on those?
The Oviedo Convention and human heritable genome editing is an interesting example. By the way, hi, Marcy. I hope that, as the pandemic fades, that you and I, 60 kilometers apart, will be able to actually see each other again on occasion. And yet the UK has authorized mitochondrial transfer, which is a form of heritable genetic modification. So yeah, there's a treaty. What does the treaty ultimately mean? how is it interpreted? How is it enforced?
Mm-hmm.
Those are tough questions. And I'm not optimistic. Marcy's more optimistic than I am about that. The 70 countries that have banned it, yes, I think that paper does show that there are 70 countries that have things in place that would prevent this at this point. Sometimes there's safety and efficacy limitations. Sometimes there's straight bans on the whole concept. There's a lot of variation one way or the other, and there's a lot of flux. So I do think that there will be countries will ban this overall. Countries will ban this for some uses. Countries will ban this, as most countries should, I think, until it's proven safe and effective. There'll be a lot of variation. I think it's not a good use of time and energy to try to get a uniform system across all the world's 200 or so jurisdictions.
Katherine?
Quick, Robin. I mean, the committee was obviously very aware of that data on the 70 countries and very aware of the Oviedo Convention, given that the Council of Europe gave evidence on it. I think ultimately the committee came down on the judgment that it's a national decision then when to move forward with the clinical applications. But also highlighted the importance of such decisions being based on technical review and societal debate. I would also be interesting to delve further into that data on the 70 countries as to ascertain why and what's the rationale behind that legislation, because there's all sorts of very interesting, sometimes people put clauses into legislation because they do it straight away without a lot of thought into it until they've explored it So it'd be really interesting, I think, to look at the 70 countries and the premise of why they've put that in there and actually which bit of legislation does it exist in. Is it actually about human genome editing or is it caught up in wider legislation? And there's just a caveat in there.
Yeah. I mean, the US is an issue there because it's a rider in an appropriations bill for one sentence, which has to be renewed every year and could easily be lost. Almost wasn't. It almost was lost 2 years ago. So, and
it's an intriguing example too, because it doesn't ban the procedure. It just bans the FDA from thinking about approving the procedure. And under general FDA law, as the FDA has interpreted it, not yet blessed by a court, it has to approve it. If Congress says it can't approve it, but as you point out, it has to be renewed every year. It's an odd sort of ban.
Mm-hmm. And of course, the UK didn't sign up to the Oviedo Convention because it was
right.
It wasn't liberal enough, and Germany didn't sign up because it was too tough,
too liberal,
too liberal. And then what if a country wants to now start using something? How easy is it to extract yourself from a treaty? So it's not necessarily a very flexible way of governance in this area.